Hi, this is Alex from devstories.eu. Scanpy Single Cell Analysis, Episode 2 of 16. The fastest way to ruin a single cell analysis is to unknowingly keep dying or empty cells in the dataset. You might think you are discovering a novel subpopulation, but you are actually just clustering cellular debris. Exposing these compromised cells relies entirely on quality control metrics. Let us clear something up right away. People often confuse calculating quality metrics with filtering out bad data. They are not the same thing. The Scanpy function, calculate QC metrics, does not remove a single cell or gene from your dataset. It is strictly an annotation tool. It calculates statistics and attaches them as new columns to your observation data frame, which tracks cells, and your variable data frame, which tracks genes. The actual removal of bad cells happens in a separate step. Why do we need these specific metrics? Consider a bone marrow sample. During extraction, physical stress can rip fragile cells open. When the cell membrane ruptures, the cytoplasmic RNA leaks out and washes away. However, mitochondria are enclosed in their own membranes, so mitochondrial RNA gets trapped inside the broken cell shell. If you sequence this droplet, you will get a high concentration of mitochondrial genes and very little else. This is a dead cell. To identify these ruptured cells, you need to track specific gene populations. In human datasets, mitochondrial genes typically start with the prefix MT-. Ribosomal genes might start with RPS or RPL. Before you can calculate metrics for these populations, you have to label them in your dataset. You do this by creating a new Boolean column in your var data frame. For example, you create a column called MT that evaluates to true if the gene name starts with MT- and false otherwise. Once you have flagged these genes, you run the calculate QC metrics function. By default, this function calculates standard baseline statistics, like the total number of RNA counts per cell and the number of genes expressed per cell. But you can also tell it to look at the specific gene populations you just defined. You pass the name of your Boolean column, like MT, into the QC vars argument. The function then computes the proportion of counts originating from that specific gene group. It adds new columns to your OBS data frame. One column will show the total counts of mitochondrial genes for each cell. Another, more critical column will show the percentage of total counts that come from mitochondrial genes. If a cell shows that 30% of its RNA is mitochondrial, you know it is likely a ruptured, dying cell from the extraction process. Here's the key insight. The calculate QC metrics function transforms raw, uninterpretable count matrices into biological signals about cell health. It does not make decisions for you, but by tagging specific gene populations, it gives you the exact numerical evidence you need to separate actual biology from extraction noise. Thanks for spending a few minutes with me. Until next time, take it easy.